Hey team, Dr. Jack Orty here, and today I'm going to take you through the T cell. Now, there's no way I could do the T cells in one video, so what I'm going to do is I'm going to give you a 30,000 foot view, overview of the T cells. It's going to be very coarse because there are many different kinds of T cells. So I'm going to give you an overview of some of the main types, and then we're going to zoom in on those types in subsequent videos and go into them into a little bit of detail. So let's jump into it here. The T cell. There is no the T cell. There's loads and loads of different kinds of T cells. So the first major division of the T cells is the T helper cell or the cytotoxic cell. And the shorthand is a TH cell or a TC cell. And they can actually be broken down a little bit more into technical detail there in terms of what they express on the surface of that T cell. So we also call T helper cells CD4 positive T cells, and we call cytotoxic cells CD8 positive T cells. And I'll go into what makes that distinction just a little bit later on in the video. Now, the T helper cells, once activated, then divide into memory T helper cells and effector T helper cells. The memory T cells are there to be reactivated should we get exposed to the same pathogen again, whereas the effector T cell goes on to do what T cells do, what T helper cells do. And the same can be said for cytotoxic T cells. They have a memory version and an effector version. So that's quite simple. This is where it gets a little bit more complicated. There are loads of different kinds of T helper cells, and this is just a few. And you might ask, why are there so many kinds, right? Well, think about how many pathogens, how many different kinds of pathogens we're exposed to, right? Imagine being exposed to a nematode, for example, which is a worm. We're exposed, there are pathogen worms, right? These are large pathogens that a single cell could not eat. They're much bigger than a single cell. It might be an intestinal worm, for example, right? Would require a completely different immune response to a virus, which is tiny and lives inside of cells, right? So you've got a giant worm wiggling around your body that requires a completely different immune response to a tiny virus that lives inside of cells, right? And so for each of those kinds of pathogens, we need a different immune response. And because a T helper cell helps mediate the immune response, we needed a different kind of T cell, T helper cell. So that's why there's so many different versions of that. So let me first just go into this division of T helper cell versus T cytotoxic cell. And what do we mean by CD4 positive and CD8 positive? So let's just jump into that. So here we have the T cell receptor, right? Now, there's a lot going on here. And I'll do a bit more of a deeper dive on the T cell receptor later on. But essentially, the T cell receptor requires these CD3 subunits to get near it in order to activate. The T cell receptor recognizes an antigen expressed by an MHC molecule. And then we have these T cell receptor inhibitors called CD45. Now, there's loads of theories about how this works, but one of the main theories about how this entire system works here is the CD3 subunits are the activators, the CD45s are the inhibitors. And in a normal situation, there's a lot of interaction between the CD3 small subunits and the CD45 large units with the T cell receptor, so it's not activated. Just in a regular situation, the membrane proteins are bumping around, and so the net effect of all this bumping and interacting is no activation of the T cell receptor. But once we recognize an antigen on this MHC molecule, what happens is the antigen presenting cell and the T cell get so close together that the long CD45 inhibitor membrane proteins are so long and so big, they get sterically hindered from accessing the T cell receptor. They can't get in there because of how close the antigen presenting cell and the T cell are. So the inhibitor cannot get in there, but the small activator, the CD3 subunits, can get in there. And once they get in there, they essentially initiate a phosphorylation cascade, which is how many receptors work, is by initiating a cascade of secondary signaling molecules. And often the cascade of secondary signaling molecules are the relationship of kinases, which is enzymes that phosphorylate things, and the phosphorylation of things. So you get kinases phosphorylating kinases, which phosphorylate kinases, which initiate a cell response, such as a change in gene transcription. That was actually probably a bit more than I wanted to go into, but once you get me talking molecular biology, I can't stop. So this is roughly how the T cell receptor works. It's at least one of the leading theories. Still a few, there's two other competing theories up in the air, but that's a major theory there. But there's actually another molecule here that needs to recognize the MHC molecule. So the T cell receptor is about recognizing the antigen presented by the MHC molecule, but we need to recognize the MHC molecule as well. And this is where CD4 or CD8 molecules come in. They're quite a long membrane protein, and they come up here and they bind to and recognize this MHC molecule. Now, the CD4 version, remember how we called them CD4 positive T cells? They are present on T helper cells, and they recognize MHC2. So T helper cells recognize MHC2, which is the professional antigen presenting cell MHC molecule. So you will find these on macrophages and dendritic cells. The MHC2 receptor antigen presenting molecule will be recognized by the CD4 molecule, which will allow the T cell receptor to come in and check whether it can recognize that antigen or not, right? So CD4 positive cells interact with antigen presenting cells, and it's the CD4 integral membrane protein and the MHC2 molecule that allow for the specific recognition of these two cell types. And then the antigen present on the MHC2 molecule will be recognized by the T cell receptor. And remember, every T cell has a unique T cell receptor that only recognizes a single antigen, and it does that through self-genetically engineering itself to have a unique gene and a unique genome, which is so cool. So cool. Now, the MHC1 molecule is expressed on all cells, right? So all cells sample the proteins in their cytosol and present it on an MHC1 molecule. Now, the MHC1 molecule is recognized by the CD8 molecule, and the CD8 molecule is present on cytotoxic T cells. So this is one of the key differences. Helper cells, MHC2 molecules, so professional antigen presenting. Cytotoxic T cells, the CD8 molecule recognizes MHC1, and so it can go up and look at an antigen on any cell in your body, and it's not looking for a professional antigen presenting, it's just looking for a cell that has sampled its own molecules inside its cytosol, and it's presented the antigen up there. But again, the T cell receptor on a cytotoxic T cell will only recognize one specific antigen because of that self-genetically engineering process that I mentioned in a previous video on B cells. Right, so that's the major difference between a T helper cell and a cytotoxic T cell. So why do they do this? Why do they have this difference? So it's activated by an antigen presenting cell using MHC2, where it's activated by any cell using MHC1. And antigen recognition by the T helper cell activates the T helper cell so it can regulate the immune response. Whereas antigen recognition on a cytotoxic T cell causes activation, which results in the cytotoxic T cell killing the MHC1 cell that it's just formed that immune synapse with, which it is just bound to. So the CD8 molecule is bound to the MHC1, sorry, and if it doesn't, if it recognizes that antigen, it will then kill the cell. So you don't want, if you're a cell who wants to stay alive, you don't want to be recognized by a cytotoxic T cell. It's right there in the name, cytotoxic, cell toxic T cell. This is just an example which I covered in the previous video on what T helper cells might do. They might release interleukin-4, which activates B cells, which then causes them to divide and produce antibodies, right? So that's an example of what a helper cell, but remember there's lots of different kinds of Th helper cells. So I'm actually going to break down the cytotoxic T cell first because it's a bit more simple. And once I've broken down the cytotoxic T cell, I'm going to go on to the T helper cells. So up in the next video, I'm going to break down the cytotoxic T cell.